And I'm delighted to be able to introduce our keynote speaker for this morning, Professor Dana Philpott, a colleague in the Department of Immunology at the University of Toronto and co-director of the Host Microbiome Research Network at the University of Toronto. Dr. Philpott holds a Canada Research Chair in Microbe-Host Interactions in Intestinal Homeostasis. She did her postdoc training and then held a group leader position at the Institut Pasteur in Paris, France, and she subsequently was recruited as a faculty member to the University of Toronto. Her research employs animal models of inflammatory bowel disease and considers how innate immunity and the microbiome shape immune homeostasis within the intestine. Dr. Philpott was a Howard Hughes International Scholar, has received an Investigator Award from the Canadian Society of Immunology and the Canadian Association of Gastroenterology Research Excellence Award. She has published in high-caliber international journals and is absolutely considered an expert in this particular nascent and emerging field of immunology. And without any further delay, let me hand over to Dr. Philpott and ask her to give her presentation. Thank you very much.
All right, thank you. Yeah, thank you so much, and thank you, Eleanor, for that nice introduction, and also to invite me for this really great conference. And thanks also to Payam and Justin for helping with all the tech loading and things. So, yeah, as Eleanor mentioned, my lab has been interested in many years in looking at how microbes interact with the host and how this affects disease outcomes, especially in inflammatory bowel disease. So I'm going to talk today about how the gut microbiome affects disease, and as I mentioned, in particular, the disease that we study, which is inflammatory bowel disease. So microbiota are associated with all mucosal surfaces in the skin, and it's really there in the environment in which they are living, which dictates the type of microbes that are associated with these surfaces. So when we think about the intestine, and in particular the large intestine or colon, we know that there are, this is where the largest abundance of bacteria are in our body. And the microbiome in the large intestine consists mainly of two phyla, the Firmicutes and the Bacteroidetes, which represent about 90% of the total bacteria at this site. And again, if you think about where they're living, this is mostly an anaerobic environment. So most of those bacteria are anaerobes. And compared to where you see the skin or the mouth, these organisms would either be aerotolerant in some way, so facultative anaerobic bacteria that can survive in this oxygenated conditions. So the microbiota, it develops, of course, we're born relatively sterile, and the microbiota changes over time as we develop and change different food, consume different foods. So at birth, the microbiota that is associated in the intestine of neonates depends on the way that you're born. So by vaginal delivery, the microbes that first colonize the intestine are typically lactobacilli, and these are the organisms that are found in the vagina. Whereas if a baby is born by cesarean section, the types of organisms that colonize the intestine are the ones that we find mainly on the skin. So as the child grows and changes different types of food, the introduction of, especially the introduction of solid food, the important part here is that the microbiota starts to diversify, and the relative abundance of different organisms starts to settle down, and the community converges to an adult-like state, probably around about 3 years of age, when the child starts to eat a diet that's resembling an adult diet. So it's really this period, this neonatal period, that's really important for the development of the microbiota, because it needs to kind of go through these different stages and build this diversity that, as we'll talk about in a moment, is very important for the resilience of the microbiota. So that's one thing many people talk about in relation to the microbiota and disease, is the capacity of the microbiota to recover from perturbations, and that's the definition of resilience. So the diversity, so the more diverse the microbiota is, the better it is to bounce back from any of these perturbations. And you can see in this graph, just as an example, food poisoning and antibiotics are like a food infection would be a perturbation that would affect the microbiota. So the adult gut microbiota is much more resilient compared to that of infants. And again, that reflects the diversity of the organisms that are found here. And lack of resilience and what we call dysbiosis is often linked to disease, and we'll discuss that on the next slide. So the dysbiosis is basically, the definition of that is an altered microbiota from its normal state. And over the past few years, many people have been studying how the gut microbiota is associated with different disease states. So certainly, dysbiosis is related to those diseases that involve the gut. So like we study inflammatory bowel disease, but also types of cancers. A lot of metabolic diseases seem to be also linked to dysbiosis of the gut microbiota, including type 2 diabetes and obesity. But we should also mention here that besides these ones that are relatively, I guess, obvious that dysbiosis might affect these outcomes, there have been a lot of studies recently that have shown that dysbiosis can also affect the behavior of an individual, so it can affect the mood and depression. But also neurodegenerative diseases like Parkinson's disease have also shown that there's some sort of microbial dysbiosis in patients that have Parkinson's. So what affects the composition of the microbiota? So some of them are listed here. So antibiotics, as we saw in that resilience slide a few slides ago, antibiotics can have quite a large effect on altering the intestinal microbiota. The diet is probably the largest way to influence the gut microbiota, and also the hygiene that individual is exposed to. So all of these changes can lead to an altered intestinal microbiota, especially in conditions where the diversity, as I mentioned, is relatively limited. And once you have that dysbiosis, this can be linked, as we talked about, to different disease states. So the disease that my lab is interested in trying to understand how the microbiota plays a role in the development of this disease are the inflammatory bowel diseases, or IBDs. So inflammatory bowel disease consists of two types of disorders: Crohn's disease, and Crohn's disease is a chronic relapsing inflammation of the entire gastrointestinal tract, so it can affect from the mouth to the anus of individuals. Ulcerative colitis is another form of IBD that's limited to the large intestine and the rectum. So both of these diseases cause a great deal of abdominal pain, diarrhea, and often weight loss. The disease is usually diagnosed kind of in the puberty range, around the age of 15 to up to about 30 years of age. And it affects both men and women equally, so there's no sex bias with these IBDs. In Canada, we're particularly hard hit by this disease. 1 in 150 Canadians are diagnosed or are living with an inflammatory bowel disease. And there are about 10,000 new diagnoses each year, so it seems to be increasing in incidence over the years. And unfortunately, the sharpest increase in IBD diagnoses are in children under the age of 10. And the incidence has actually doubled in Canada and the UK between 1995 and 2005. So again, it seems to be something that is continually on the rise in our society. So thinking about the possible effect of microbes on the development of these inflammatory bowel disease. So there have been many studies over the years that have looked at the microbial composition associated with inflammatory bowel disease. And this is just one example. There's, like I mentioned, there's many studies that have looked at this. And this is a study by Frank et al. That was published quite a few years ago now, 2007. But basically, they show relatively the same kind of information that in a patient that is suffering from IBD, we have an expansion of these proteobacteria. So these are usually facultative anaerobic bacteria that can grow in environments that have oxygen, which we think happens when you have inflammation. The level of oxygen within the intestine probably increases. There seems to be a contraction of certain types of taxa within the gut microbiome. And one of the interesting findings that has been continually shown is this absence of this organism called Faecalibacterium prausnitzii. And it seems like this bacteria in vitro studies and some in vivo studies seems to have some protective effects against inflammation. So this bacteria consistently seems to be, the species of bacteria seems to consistently dropped out of the microbiota in cases of inflammation. But one question, the problem with these studies is this the chicken or the egg? We know that inflammation drives dysbiosis. As I mentioned, when you have that kind of elevated oxygen levels within the intestine, that's going to change the relative abundance of certain organisms. So what we're really interested in is trying to understand if this dysbiosis is actually an indication of the development of disease. So the only way we can really look at that is to try to look at the gut microbiota before disease onset. And one of our colleagues, Ken Croitereau here at the University of Toronto, has a very extensive study going on at the moment looking at the gut microbiota of individuals that don't have IBD yet, but they are susceptible to IBD. So they have a first-degree relative that has an IBD. So hopefully with those studies, we might be able to see that there are some organisms that can predict the development of these diseases. So Crohn's disease in particular is the one that we study in my lab, and it's, as we know, multifactorial. So we know there's a role for the microbiota. We know there's some sort of dysregulated immune response that's associated with Crohn's. There's environmental effects. For example, smoking can trigger Crohn's in susceptible individuals. And finally, genetics play also a large role. From the, I think now there's about 180 genetic loci that have been associated with Crohn's. Nod2, which is the one that we study in my lab, has the strongest genetic link to the development of Crohn's disease. So I'm just going to introduce a little bit about Nod2 so I can tell you about the study that we're doing at the moment. So Nod2 is a pattern recognition receptor of the innate immune system. So it's able to detect fragments of bacteria, and in this case, for this particular pattern recognition receptor, that's peptidoglycan, which is part of the cell wall of all bacteria. And specifically, it recognizes a fragment of peptidoglycan from a bacteria called muranyl dipeptide, or MDP. So once MDP interacts with Nod2, its activation leads to NF-kappa B and MAP kinase mediated gene expression. And the mutation, an interesting mutation we showed a few years ago that's associated with Crohn's disease, results in an inability to sense MDP. And this is just a cartoon of the Nod2 molecule. So it has this leucine-rich repeat domain at the C-terminus, where we think this, where we have some evidence that MDP can bind directly to this portion of the molecule. And in individuals that have Crohn's, they have this frameshift mutation that I indicate here, that again results in a molecule that's no longer able to detect this molecule. So we know, so that's kind of the genetic side of things with Crohn's, but we know that there are a number of environmental factors that are likely critical for the development of Crohn's. And we know that because there are individuals that have these Nod2 mutations that don't develop Crohn's disease, so it's not 100% causative, as well as the fact that we have all sorts of Nod2 deficient mice in different tissue types and whole body knockouts. And these mice do not develop spontaneous disease. So you really need a trigger in order for those mice to develop disease. So of the environmental factors that might trigger disease in the background of Nod2 mutation, we look at the role of the microbiota with a specific question whether dysbiosis can that influence Crohn's disease in a genetically susceptible individual. So this is our hypothesis. We hypothesize that if we alter the microbiota, and we do this using a short-term treatment of antibiotics within the neonatal period, and as I mentioned, the reason why we choose this neonatal period is that the neonatal period is a time where the microbiota is still kind of in this chaos. It's still trying to figure itself out, and it's not very resilient to different perturbations. So we wanted to perturb the microbiota at that time point, and then look at how this might affect disease development in a Nod2 deficient host. That's the picture, I guess. The antibiotics affecting the microbiota and how this might affect the development of disease. So the experimental questions we have is, first of all, does antibiotic treatment during this neonatal period alter the gut microbiota composition? And do early life antibiotic exposure increase the susceptibility to colitis in this mouse model? So the way we did this experiment is we wanted to use kind of a low dose antibiotics, which would kind of mimic what a human neonate would be exposed to if they had some type of infection. And what we did is and this was with amoxicillin, we put this into the drinking water of the dam before she gave birth to the pups. So the pups were exposed from day 0, so from the time they were born, to antibiotics that were present in the milk from the mom, up to weaning, and then the mice were then switched on to normal water. So it was a relatively brief and low dose antibiotics during this time point. I'm not going to go through this in detail. Hopefully some people know how we understand how to assess the composition of the microbiota. So basically, we look at sequencing of this 16S rRNA gene. So this is a gene that's found in all microbes, and there are conserved regions and variable regions. And by sequencing those variable regions of the 16S rRNA gene, we can put those organisms into different classifications. And using 16S, we can usually go down as low as families, so we can put taxa to different families, and sometimes up to genus, but it depends on really whether those databases that are out there have this kind of information in them. So when we use 16S rRNA, again, we can look at the composition of the microbiota within different contexts. And the first question we needed to address was whether Nod2 genotype, does that actually influence the murine gut microbiota at baseline? So this is all without any perturbations. And when you look in the literature, there have been many studies looking at the influence of Nod2 on the microbiota, and there's been all sorts of studies. But I think if you do it correctly, and that is the use of littermate mice, then you can really have an idea of whether that gene is influencing the composition of the gut microbiota. So the way to do these kinds of experiments properly is to use het mice, so het Nod2 mice. So then you get the full complement of wild-type het and knockout animals. And this using littermate controls is really important because it controls for maternal transfer of microbes, and also, very importantly, the early life environmental influences that might affect those microbiota. So I'll just, if anyone wants more information on littermate mice and analyzing microbial composition correctly, we wrote a review a few months ago in Trends in Immunology on this. So using that kind of an approach, we did 16S RNA sequencing of the gut composition from these different genotypes. And when you use principal coordinates analysis, and in this case, we used what's called the Bray-Curtis dissimilarity test. And this is just a measure of similarity among different groups. You can see that basically the het, wild type, and knockout mice kind of scatter, and there aren't any particular groupings. So this indicates that there is no difference in the microbiota between these genotypes. And that was confirmed when we also looked at the relative abundance. So we looked at the actual taxa that were making up these microbiota. And there was no difference between the three different genotypes. So that's at baseline, at least, there are no differences in the microbiota. And now we can go then and perturb that microbiota and see what happens. So we did that. We did our neonatal antibiotic treatment of Nod2 knockout mice and their wild type littermate controls. And again, using this principal coordinates analysis, we were able to look at the composition of the microbiota in these different conditions. This was at day 35, so this was about a week or I guess almost 2 weeks post the cessation of the antibiotics. So we're really looking at the resilience of how is the microbiota coming back over time. And you can see that most of the microbiota is kind of clustered in this little quadrant here, but the Nod2 knockout mice with antibiotics were the most affected by the antibiotics, and they weren't coming back to their normal state. So this is a significant difference compared to the other groups. And then what we wanted to do is to try to understand what are the organisms that are making up the microbiota that reflect that dissimilarity among the groups. And the way we do that is using this another statistical analysis called the Lefse analysis. And this enables us to compare between experimental groups to determine which bacterial either families or genera discriminate between these two groups. And doing that, we saw that the Nod2 knockout mice with antibiotics, they had more of this family of bacteria within their microbiota, and they were missing these ones that are shown here in blue, which were more associated with the wild type mice on antibiotics. So there seemed to be some organisms that were different and kind of popping out in the Nod2 knockout mice, and some were actually missing. So the next question is that does this dysbiosis that we've induced lead to more severe disease? So the mice then, after the antibiotic treatment at 56 days of age, these mice were treated with DSS. And this is a chemical that we put in the water, and it's like basically just strips off the epithelium of the intestine. It's rather harsh treatment, but a very accepted model that people use to study Crohn's. So we have both the antibiotic and water-treated wild type and knockout mice. And what we found is that the mice that had the neonatal antibiotics, but only the Nod2 genotype, these mice had more severe disease, so they lost more weight, shown here, compared to the other groups. And they also had a greater degree of inflammation. Here we're measuring lipocalin, which is a protein that's produced by neutrophils and is a very good indicator of gut inflammation in our model. And again, so the Nod2 knockout mice on antibiotics had a greater degree of inflammation compared to the other groups. And this is also consistent with what we saw in pathological assessment by our pathologist, Dr. Kathy Struker, where she saw that the Nod2 knockout mice on antibiotics had a greater degree of pathology compared to those wild type animals. We then wanted to look at how the antibiotics are affecting the mucosal immune environment, and maybe this could explain how or why we're seeing greater degree of inflammation in these animals. Now, I'm not going to go through a lot of details because I'm going to go over time, but basically the neonatal antibiotics in both wild type mice and in Nod2 knockout mice perturb the proportions of T cell subsets within the gut. So here we're looking at different T cells, including the RR gamma T T cells, the FOXP3 T regulatory cells, and these RR gamma T Tregs. So all of these seem to be differing in their proportions under treatment with antibiotics. So that was kind of an interesting finding. And the fact that the mice that were on antibiotics, especially the Nod2 knockout mice, when we treated them with DSS, had a greater degree of inflammation within that environment when we looked at different inflammatory cytokines like IL-1 and here IL-33. So the antibiotics seem to create a more inflammatory environment, and we want to see in subsequent studies if this is really driven by this disproportionate T cell subsets within the gut. Okay, so the next final question is then, is this dysbiotic microbiota sufficient to increase disease susceptibility? And the way we wanted to test that was then to use our same setup that I just showed you in the previous slide, so with antibiotic treatment during the neonatal period. And then we took the stool samples from those mice and transferred them into germ-free Nod2 knockout mice to look at whether these antibiotic-treated microbiota was sufficient to drive increased susceptibility in those animals. So we took, we did the microbiota transplant at weaning into germ-free Nod2 knockout mice, and then again performed this colitis experiment later in life. And interestingly, we found that this microbiota was sufficient to drive increased disease in this background. So the mice lost more weight. So the mice that received this antibiotic-treated microbiota, shown here in pink, those mice had more severe lipocalin levels, so more inflammation in the gut, and their colon was also shortened, which is another indicator of inflammation within the intestine. So this microbiota from those neonatal mice that were treated with antibiotics promotes disease when you re-put them into a germ-free animal, in a Nod2 deficient germ-free animal. So then in summary, we showed that if you have an altered microbial composition, it seems to influence the immune environment within the intestine. And we think this is really critical for the susceptibility to disease. Mucosal damage and colitis can be altered through microbial manipulation. And something I didn't mention, but the timing of the antibiotic treatment is really key, because if we did the same experiments in adults, so adult treated with antibiotics, we did not see the same effect. So it's really a particular time point within that neonatal period where the microbiota is less resilient. So if you have antibiotics during that period, the microbes have a hard time coming back to normal. And we think that this is important for driving colitis. So in the future studies, as I mentioned, we want to understand whether it's really this T-cell homeostasis within the intestine of animals that are exposed to neonatal antibiotics. Is that really what's important in driving disease susceptibility? And on the other side of things, we also want to try to identify and test organisms or group of organisms that might be responsible for this increased susceptibility. So we're trying to culture out those different microbes and then put them, re-put them back into a Nod2 germ-free environment to try to see if we can really understand if one or a few groups of organisms are really driving this susceptibility. So that might be a way that we can alter the microbiota in the future to protect people from the development of Crohn's. So I'll end there and thank the people that did this work. So this was a collaborative project with Dr. Ken Kreuter's lab, and this our PhD student that we co-supervised, Ashley Gettle, Susan Robertson and Williams Turpin did all the microbiota analysis for this study. So I'll end there and thank you all, and take any questions.
Thank you very much, Dr. Philpott. And just wanted to let you know how wide the audience is. I'm going to read off a list of countries where we have participants online right now who heard your talk. So there were participants from, hold your breath, India, Brazil, Bangladesh, the UK, Kenya, Portugal, Afghanistan, Kazakhstan, Russia, Nigeria, USA, Indonesia, Burma, Malaysia, Finland, Australia, South Africa, Iran, Colombia, Argentina, Poland, and Germany. So I really do think we have an amazing reach.
That's incredible. That's really
What
nice.
I'd like to do now, if you've got a few moments, Dr. Philpott, is ask you a number of questions that have popped up on the chat line.
Sure.
And perhaps you can answer them live for us. So one of the questions was, do different antibiotics affect the severity of the DSS-induced colitis? And is this reflected in different profile of T-cell subsets?
That's a great study that we were thinking about doing in the future, is try to see if, you know, we looked at amoxicillin, but trying to look at maybe antibiotics that have a specific sensitivity towards certain microbes. Maybe if we wipe out a certain type of microbes, maybe that's the ones that are either protective or driving disease. So that's, again, this is something in the plans, in the works, seeing how different antibiotics might affect the dysbiosis and how this might drive disease. So good question.
And have you had an opportunity to look at the viral composition to see what the influence that might be?
Yeah, so that's a huge drawback of all these studies. I mean, we talk about the microbiota, but we focus in on bacteria only, right? And we know from some of our previous work, we had a parasite that was having a huge effect on T cell homeostasis within the gut. This is first described by Arthur Motha in our group. And this parasite, you know, it completely flips your T cell responses within the gut. And so, yeah, if people aren't careful and not looking at this, then you don't know if it's whether it's the bacteria within the microbiota or other organisms. And indeed, like viruses and bacteriophage are another component that people are looking at, but we haven't yet gone down that route. It's a lot more difficult to find or assign these sequences to viruses. But I definitely appreciate the question that we're really, you know, only focused on microbes and ignoring all the other kingdoms out there. Right? So, good point.
So another question was whether you have any information about the small intestine and which immune subsets might affect changes in microbiota. Yeah,
that's also a particular area we're very interested in because Crohn's with Nod2 especially is associated with ileal disease. So many of our studies are looking at how the ileal microbiota influences development of inflammation within the site. It's a little harder to look at most models of ileal disease just because there's fewer types of models out there. We use this what's called anti-CD3, so we just blast the T cells in the small intestine, and you get this very acute inflammation. And so that's one of the ways that we've been looking at the small intestine. But again, I appreciate that. Yeah, we're focused on the colon, and in many cases, it's probably the ileum that should be studied, and the ileal microbiota. I mean, it's something else that many people don't look at because we sample the stool, which is a reflection of what's going on in the colon and the cecum of these animals. And you know, sampling the ileum is a little bit more challenging in most models, but something, yeah, again, we're really interested in following up on. So.
Another question: what's the relationship between Nod2 mutations and IL-10 production in the context of Crohn's?
That's yeah. So a lot of groups have looked at the IL-10 levels in Nod2 knockout mice, and I think, well, I guess in the study that I just showed you, we did see a little bit of an alteration in IL-10 in the Nod2 knockout, but it wasn't striking. I think there was just a trend to more IL-10 less IL-10 in the Nod2 knockout mice. But you know, for sure, it's an extremely important cytokine, an anti-inflammatory cytokine that would be key in this disease. And yeah, we need to look at that more for sure. Good point.
Okay, well, one last question before we allow you to go and have your cup of coffee is presumably different animal facilities will have different environments in the context of exposure to bacteria. So does the incidence of DSS-induced colitis in the Nod2 deficient mice vary different facilities around the globe?
Wow, whoever asked that question, you're hitting the nail on the head. And that's one of the huge problems we have at the moment is the variability from facility to facility, and even within facility variability. And it's going to be something really hard to deal with. I mean, one idea that we had is that maybe everyone needs to start with a similar microbiota. So maybe, you know, we have like a on-the-shelf microbiota that everyone would use for these types of studies. So you colonize your mice with this microbiota, and then hopefully reproducibly you would see the same sort of inflammation in the different facilities. But Yeah, if you want to have a look at the review that we wrote in Trends in Immunology, we talk about that problem. Because yeah, from one facility to the next, you see varying degrees of inflammation with this model. So it's very difficult to assess. One of the proposals that we also have in the Trends in review is to verify results in different facilities. So link up with a colleague and do the experiments to show that that phenotype is really penetrant also in another facility. So good point.
Thank you very much. So from all those, I've just been told there are 2 more questions. Let me just get to those 2 more questions. Oh boy, bear with me because I just clicked out. All right, so 2 more questions. I haven't got 2 more questions. Oh yeah, one of them is about any thoughts on Treg homeostasis in Crohn's or allergies, and are the ILC subsets affected by the early antibiotic treatments?
So we have not yet looked at ILCs in this model. I think it's really the beginning of this kind of early life perturbations. It's kind of a theme that we're really exploring in my lab at the moment. Early life perturbations, whether it be antibiotics or early life infections, another thing that we're looking at, and trying to understand that if this has any influence on ILCs, but also Tregs and, well, I guess it's just different proportions of T cell subsets within the gut. So that's something that we're definitely following up on with these models. Yeah.
All right, so I think we'll end it at that point. Thank you very much again for a great presentation, and I'm going to hand off now to Ash, who's going to take over for the rest of the talks. Thank you very much, Dr. Philpott.